Hello everyone, thank you for coming. My name is Carlos Ramirez and I'm a PhD student at Katie Paco's lab in the University of Bern, Switzerland. Today I'm going to tell you a little bit about the work I'm doing on the molecular mechanisms behind EDA-mediated adaptation to fresh water in tree spine stickleback. One of the main goals in evolutionary biology is to understand how organisms adapt to new environments. Adaptation to new environments often requires changes in many traits in response to differences in abiotic factors like temperature, pH, humidity, and chemical composition, and changes in biotic factors like predation, parasites, and prey. One of the main questions in this field is what are the genetic basis of these adaptations? What are the genes involved? Do these genes have pleiotropic effects on multiple traits? And how are these pleiotropic effects mediated at a molecular level? However, it's difficult to answer these questions because most organisms don't have the necessary genetic tools to answer them. That's why tree spine stickleback is such an important model. It is one of the few examples of adaptation to new environments where we have the necessary genetic tools to answer these questions. Around 15,000 years ago, after the last glacial maximum, many new freshwater environments became available on the northern hemisphere, and tree spine sticklebacks migrated from the ocean to colonize these new freshwater environments. Even though these colonizations happen independently, freshwater populations often show parallelism on the adaptations that they have evolved. For example, in the lateral plates. As you can see in this figure, while marine stickleback tend to have armor plate from head to tail, freshwater sticklebacks lose half of those plates and only have plates from the head to the middle of the body. We already know that the major locus involved in this loss of lateral plates is ectodysplasin A, also known as EDA. In vertebrates, this gene is involved in the development of epithelial appendages like hair, teeth, and scales. And in tree spine stickleback, this gene is found within a 16 kb haplotype that is conserved in freshwater populations. This haplotype includes two immune genes also, TNSF13B and GARP. And at least in the population I'm working with, the marine allele for complete plates is dominant over the freshwater allele for low-plated individuals. We also found recently that EDA is actually a pleiotropic gene, affecting also the sensory lateral line patterning and even schooling behavior. However, we still know very little on how EDA works to achieve these phenotypes. We don't know what is the main causal mutations for the EDA phenotype, nor which are the gene pathways through which EDA operates. Trying to shed light into these questions is the purpose of my research. For today's talk, I will, however, only focus on the second project. In this project, we want to know in particular two things. What are the genes affected by the EDA haplotype, and whether are there other cryptic molecular phenotypes of the EDA haplotype, for example, immunity, which could explain why we have two immune genes in the haplotype. In order to tackle this question, we use transcriptome sequencing. There are already studies comparing the transcriptome of marine and freshwater sticklebacks, but no study so far has tried to disentangle the effects of only the EDA haplotype. And this is what we try to do in our study. In order to disentangle the effect of the haplotype only, we use the following experimental design. We look for heterozygous marine individuals that happen at low frequency in nature. These individuals look phenotypically marine, but they actually carry one low-plated EDA allele. If we cross these individuals between them, then what we will get are offspring that are mostly marine, except at the EDA haplotype, where they will have all the three different combinations or the three possible EDA genotypes. This is what we did, and then we selected 4 individuals from each one of the EDA genotypes. We extracted skin after plate development and head kidney, because head kidney is the main immune organ in fish, and we sent them for transcriptome sequencing. Then, once we got the data, we used the following bioinformatic pipeline, where we started by refining the quality of the reads coming from the sequencing center. Then we mapped these reads into the genome to find out from which genes each read came from. This allows us then to go to the third step, where we went to each gene and measured the gene expression of it by counting how many reads mapped into that gene. Then, with this read count data, we were able to perform differential gene expression analysis to find what genes were differentially expressed between genotypes. And we found that there were hundreds of differentially expressed genes. Remember, these are fish that, apart from the haplotype, they all have the same marine genomic background. So all of these genes that we found differentially expressed should be because of the haplotype. As we would expect, the great majority of the genes differentially expressed was found in skin, especially in the comparison between CC and LL individuals. But we also found some differentially expressed genes in head kidney, though very few. So we focused our analysis mainly on the skin results. Also, confirming previous findings, we found that EDA itself was differentially expressed in skin. We then did a functional enrichment analysis of the differentially expressed genes in skin, and this showed that this wasn't just a random set of genes. They were actually biased towards certain pathways. Especially what we found was that there were many genes involved in uh, development and bone ossification, exactly what we would expect to be the genes involved in the development of the lateral plates. Very interestingly too, our gene set was also enriched in genes related to neuromuscular process controlling balance and netrine activated signaling pathway. The netrins are proteins involved in axon guidance, and we think that these two neuronal terms that we found to be related to the effects of the haplotype and the patterning of the sensory lateral line. Despite having found these promising results in the differentially expressed genes, we decided to take our analysis a bit further and look also to another mechanism of transcript regulation, alternative splicing. Just as a brief reminder, alternative splicing is a mechanism through which from a single gene, multiple messenger RNAs are created. This happens by excluding and maintaining different combinations of exons during the maturation step of pre-RNA into messenger RNA. Several recent studies have found that alternative splicing can play an important role in phenotypic evolution and adaptation. Thus, we decided to see whether the haplotype would affect the alternative splicing of other genes. And we were able to test this with basically the same pipeline that we use for the differential gene expression, just that instead of testing for differential gene expression, we tested for differential exon expression. And we found that there were evidences for alternative splicing of dozens of genes. Not as many as there were differentially expressed, but for example, in some cases, like in head kidney, we had more different alternatively spliced genes than differentially expressed ones. A quick look at the top differentially spliced genes in skin identified genes related to bone differentiation and neuronal development, just like in the differentially expressed genes. However, when we compare the two sets of genes, we found them to be mostly non-overlapping, meaning that differential expression and alternative splicing are mostly independent mechanisms, probably used in different contexts, and alternative splicing is not some sort of side effect of differential expression or vice versa. So in summary, what I found so far in my project is that the haplotype affects the gene expression and or alternative splicing of hundreds of genes, even when the rest of the genome is the same. And these two regulatory mechanisms seem to be most independent. Also, the genes that we found had functions related to bone and neuronal development, suggesting that they could be involved on the phenotypic effects of the EDA genotype, haplotype, sorry. So Going from here, the next steps that we are planning to do are to look more in depth into this list of differential expressed and spliced genes, try to identify exactly what genes they are, what pathways exactly they belong to, whether they are from the same pathways or not. We want also to validate the most important genes with real-time PCR, both for the differential expression and for the alternative splicing. And finally, do co-expression analysis to infer gene interactions and whether these are affected by the EDA haplotype. So to conclude, I would just like to uh, thank a lot to the people with whom I work every day. All of them, directly or indirectly, help a lot to make this work possible. And a special thank, of course, to my two great supervisors, Professor Dr. Catherine Peichel and Dr. Araxio Rutia, without whom this work would never be possible. I would, of course, also like to thank to the EMPSEB 26 organization for giving me the chance to talk to you about my work today, and also thank you for your attention. If you have any question, feel free to ask. Thank you very much.